Hey, hello everybody. This is a video for March 26th, and you can also review this on March 27th. I'm going to be giving you some questions to answer tomorrow. I had some difficulty on Google Forms with that last quiz. I saw that you all turned it in that did it, but it wasn't able to show me your score. So I'm going to have you today and or tomorrow when you're going to do these questions, I'm going to have you go to this website. It's called ck12.org and / for student, and then you're going to click up here to join. I'll show you the class code in a minute, but this is something I've used years before when we were in the computer lab. It's a good way to have some review of some information and also for me to ask you some questions. So if you click on join up here, it should send you to a page where you can sign up with Google. So after it loads here, hopefully it loads soon and I can show you, but let me for now show you the class code and I'll come back to this one. So if you look here on the CK12 website here for the dashboard, I did one for all the different class periods. So period 1 is here, there's the class code 5XJ04. And then period 2, it's an LF1TK. And then period 3, yours is WYA3U. Period 4, N4MDQ. Period 6, LYMXH. And then this should be life science period. I did period 1, 2, 3, 4. And the other one, I'm missing the math one. I'll put the math one up on the next video. There it is. The math one is N2MV5. This other one, oh yeah, this one over here is for all the classes. I'll probably be posting a lot of the stuff in your period class because it will make it easier when we go to give you your grades, so I know who was in which class. But this one here will be for all classes, but I'll be using more so the ones for your specific class period. But I'll give you this one as well, B2Z3U. As you see with the class codes, it kind of has a similar setup to Google Classroom, in which it has the class codes and classes to join. But it just has a lot more stuff for science and math on this website. So it's ck12.org. Make sure you don't do .com. Make sure you do ck12.org. And let me see if the other page loaded for you to sign up. It doesn't look like it has loaded still. I'll try to refresh it. But if not, hopefully it will show up on your computer. There it is. So when you do this, it should say student sign up. Don't do the teacher one. And you should just be able to click sign up with Google. And it should open a new window. And it should have a place for you to put your information. So put your school district email. Remember, it's the way you sign into the computer. And then the @montebello.k12.ca.us. And then just put in the front part of your email here in the front with your initials and everything. And then for your password, I suggest you just use the same password you always use, the first 5 of your last name to make it easier, or your school loop password when it tells you to do that. And then when you get in there, you'll be able to click on join a class once you're signed in. And you'll be able to, kind of like in Google Classroom, it won't let you do it now because you're not signed up. But you'd click on join, and then you would join the class. And then I'm going to post a set of questions tomorrow to review. So back to this here, I'm going to show a video on meiosis. And I'll be kind of pausing the video at points because it's going to show this whole process of meiosis with the productions of these sex cells with half the number of chromosomes, as it says up here. So it's going to go through this process in the video. You should read through this part down here about meiosis and write down any notes that you need to do. But I think the video will help you to understand it, and I know some of you had questions on the lesson yesterday as well. So here's the video.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells,
So remember, I went over this a while ago. You might not remember. It's been a very long time. But just to review, remember, mitosis is just copying those body cells. So if you have stomach cells, skin cells, hair cells, whatever they might be, they're going to be making exact copies. Meiosis is going to be a similar process with the cells dividing, but it's going to be slightly different.
Meiosis is important for growth and for repair of damage, or to replace worn-out cells, but not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes.
Remember, it's going to be 23 on each of these because half will be from the mother and half will be from the father. So they're going to have the 23 for each one, and they're going to combine together for an offspring with the 46 pairs. If you remember from the book, the example with the grasshoppers, same kind of process, but remember with the grasshoppers, it's going to be 24 chromosomes and then 12 for the sex cells because they're going to be combining together.
Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually,
I'm just going back real quick because I know we have some twins in our classes. Hello, twins and everybody. So remember, when this happens and there's a sperm cell and egg cell, different ways that different twins can form. One way is with identical twins, which we have pairs of in our classes. The egg cell, after fertilization occurs, the egg cell can split and it can form from the same egg and sperm cell the identical type of twins. However, there's other types in which there's going to be 2 egg cells and 2 sperm cells. Those would be called paternal twins. That would mean that that's why there could be one male and one female, even though they're twins. The one sperm cell going into the one egg could be carrying a Y chromosome, and the other one going to another egg could be an X chromosome. That's why sometimes twins can be male and female, not always identical.
Egg cells have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis you're going to divide twice, and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before. And it kind of helps to remember that it comes before. Chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1 where you have two newly formed nuclei and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1 though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over that doesn't happen again in prophase 2. You have your chromosomes and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful as having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study.
Okay, so review the video, watch it at least once, write down notes, and also as you're watching the video, if you have your physical book, be looking at your physical book or have another window open here to show the different steps that were animated in that video. Tomorrow, make sure you log into that website, the ck12.org, put in your class code, and I'm going to have an assignment up for you to complete answering questions. If you need help, email me on School Loop, and thank you.